Welcome back. Hopefully you can hear me a little better now. I realized that while I've been wearing this very stylish headset, I've actually been recording off the microphone on the webcam, which is why the audio quality wasn't all it could have been in the first couple videos. So I apologize for that, but hopefully you can hear me now. So in this chapter, we're going to talk about functional neuroanatomy. So the neuroanatomy of the nervous system, and also about how it affects behavior. So in this lecture, this mini lecture, we're going to actually be discussing the anatomy of the nervous system. So talking about neurons, the parts of neurons, and other cells in the nervous system that are important. So neurons, also known as nerve cells, are the most important part of the nervous system because they're the basic units of the nervous system. Each one includes a cell body, like any other cell, but they also have some things that are specific to neurons. They have receptive extensions called dendrites, a transmitting extension called an axon, and there are a ton of them. It's estimated that there are between 100 and 150 billion neurons in the brain and the nervous system. So there are tons and tons of neurons. In addition to neurons, you also have glial cells, and these are the support cells for the neurons. They provide structural, nutritional, and other types of support to the brain. And they're also very important for myelination, which we'll discuss in this lecture. It was debated early in the history of biological psychology whether each neuron was independent or whether they created a long chain that was unbroken, like a really long neuron. And this was actually settled based upon the work of Ramon Cajal, a biologist who helped us come up with the neuron doctrine, which is he used an electron microscope and essentially was able to see the little gaps in between the neurons, which were later named synapses. So finally settling this argument on whether or not the brain is made of independent cells or these very long neurons, we found that no, they're actually independent cells with little gaps in between them. And that information is transmitted from cell to cell through these synapses, as we'll talk about in the coming lectures. So the neuron has several structures that are common with any other cell. They have mitochondria, so these are the power systems in the cell. They produce energy. There also is a cell nucleus, which contains the genetic code for the neuron. So much like any other cell, it has a nucleus. And there are also ribosomes. And these are the components that translate genetic information in order to create proteins. So in some ways, neurons are like any other cell you would see. However, of course, they're different. As we mentioned, there are those dendrites and also the axon that differentiate neurons from most other cells. So there are 4 zones you need to know about in reference to the neuron. The first is the input zone, and that is up here. That's the dendrites. So the input zone receives information from other cells through the dendrites. So this is where information from other neurons comes to the neuron. So that's the first part. There's also the integration zone, so that's here at the very base of the cell body. And this is the part where all the messages are combined and transformed. So basically, this is the place where all the either excitatory or inhibitory messages are brought together, and it's determined whether there's sufficient stimulation to cause the neuron to fire. As we'll be discussing, the neuron is an all or nothing thing. So it fires or it doesn't. So it's figuring out, is there enough stimulation to cause this firing? The conduction zone is down here in the axon, and that's where the single axon leads away from the cell body and transmits that message into a chemical message, or a electronic message rather. I apologize for that. Electronic impulse that travels down the axon and goes to the last zone, which is the output zone. So the output zone is where the information is then transmitted to other neurons. So we'll see how this works as time goes on, but output zone is where the axon connects with other dendrites and sends that message along to the other neurons. And this is done through releasing chemical messengers called neurotransmitters into the synapse to pass that signal along. So neurons are classified in many different ways, but primarily by their shape, their size, and their function. So multipolar neurons are the standard neuron that you see drawn. This is one axon, many dendrites, and this is the most prevalent type of neuron. And you'll see this on the next slide. So don't worry about trying to picture these. Just feel free to write down the information. So multipolar, the most common type of neuron, one axon, many dendrites. You can also have bipolar neurons. Bipolar neurons have one dendrite and one axon, and these are very common in sensory systems such as vision. This is very good for when you need to just copy something that's in it from 1 to 1 to 1 to 1 to 1. And now when you're trying to gather a lot of information together in order to figure out whether or not you fire. So those are bipolar neurons. And again, they're common in sensory systems. There are also monopolar neurons, which have a single axon that branches in two directions. On one side, there's an input zone with branches like a dendrite, and on the other end, there's an output zone. And these cells often transmit touch information from the body to the spinal cord. So what you notice is that multipolar neurons are by far the most common, and these other ones, the bipolar and monopolar, have specific uses or specific functions that they're responsible for, and that's part of why they're different. So here, what you see is this is a multipolar neuron. So this is the one that's the most common. You see, throughout the brain, many dendrites, one axon leading away. Here's the bipolar neuron, where you have one dendrite and one axon. And so, shaped in much the same way, what you see is, with both of these, the cell body is in the same place, the integration zone is in the same place. And I bring that up because unipolar neurons are different. As you see, unipolar neurons have this setup where you actually have a single axon that branches in two directions. So this is all axon. And for this all to be axon, that means that this integration, this deciding whether or not the cell fires, needs to happen earlier. So that's what you see. You have the dendrites up here. You have the integration up here, outside of the cell body, which is uncommon. And then it shoots down the axon like this. So again, you see these a lot with sensing touch information. So those are the three main different types of neurons that you'll need to know about. Also, there are differences in size. Larger neurons are often more complex. They usually cover more distance, and they're also somewhat faster at sending information. And I say somewhat faster because overall there's a general rule that a larger neuron will send information faster. The caveat being, it depends if it's myelinated or not. If you're just looking apples to apples, unmyelinated or myelinated, larger is faster. But if you have a smaller one that's myelinated, that's going to be significantly faster than a larger one that is not myelinated. So neurons can also differ in their function. Motor neurons are neurons that transmit information to muscles or glands. Sensory neurons, as you may guess from the name, go to the receptors, or from the receptors rather, to the brain and respond to environmental stimuli, such as odor, light, touch, things of that nature. And then there are inner neurons. Inner neurons are the vast majority of neurons. So these receive input from other neurons and send input to other neurons. So they kind of connect the system. So most neurons are these inner neurons. And again, what we kind of see is where the generic neurons are the most prevalent. And then there are these two others that have specific uses, and they're a little less prevalent, well, much less prevalent, but that's the difference, is the function. Then we have glial cells. These are a whole different type of cell, which are really fun, I think. Glial cells are the support cells for the nervous system. So the neurons can't do it alone. The neurons manage the information basically, but they need the support cells in order to keep them up and running. And that's what glial cells do. They help provide raw materials, aid in neuronal communication, and they do many other tasks. And there are only 4 basic types of glial cells, so there are 4 types that you'll need to know. First are astrocytes. Astrocytes are star-shaped cells that support the neuron. They connect with capillaries to help regulate blood flow, and they also provide raw materials to the neurons, such as what you see in this picture. So here, you see this is a capillary, and you have this astrocyte connecting to the neurons. So it's providing blood and nutrition, so all the things that the neuron needs in order to stay alive. There are also microglia, and microglia are the brain's cleanup crew. They are small, as their name suggests, but they're very active. Anytime there's an injury, they go to the injury and they remove any debris from the injured neuron. Also, I want to jump back to astrocytes for a second. A couple other things with astrocytes. Astrocytes also will connect with other astrocytes and neurons in order to help regulate the system. So one thing that they do is they actually help connect the neurons in more of a network so that they're all working together. They also will help in the formation of new synapses, so helping guide neurons together. So astrocytes are very, very active as well, not just in maintaining the neurons, but also with broadly maintaining the network. So here you just have a bigger picture of a astrocyte connecting between a capillary and a neuron, and you also have microglia down here, much smaller, who will take debris and get rid of it. So myelination is an extremely important process in the nervous system. It's the process by which glial cells wrap axons with a fatty sheath that we call myelin, and this insulates the axon, and it makes for a much faster transfer of information. We'll get into this a lot more later, but much faster transmission. So with this, so you have the axon. Let me actually grab a prop. So I have my axon. So myelin kind of looks like this, where it's covering parts of the axon. Now there are parts in the middle that are not covered. Those are your nodes of Ranvier. They are the gaps in between sections of myelin where the axon is exposed. And we'll get more into that as well later. And in multiple sclerosis, you may have heard of this disease. This is a demyelinating disease. So what happens is that myelin that you have starts dying off. You start losing it. So the connections in your brain become much slower. And that really plays havoc throughout the whole nervous system. So how does this happen? How do we get this myelin? Well, there are a couple glial cells that are responsible for this. So the first one is Oligodendrocytes, and these are glial cells that form myelin sheaths in the brain and the spinal cord. So these are cells that are much smaller than astrocytes, but are bigger than microglia. And what they will typically do is contribute sheathing to numerous adjacent axons wrapping around the axon. So you can have one oligodendrocyte that's grasping onto several different axons in the neighborhood. This process takes some time, as you might imagine. We think that the whole process of myelination may take, well, it's debated based on who you ask, but it may take 10 to 15 years after birth. Some say longer. Some say, maybe even mid-20s. But the long and the short of it is this process takes some time. And actually it should help explain why children are sometimes uncoordinated, but that improves with time. Because also our ability to send information in the brain improves with time. Then there are Schwann cells. Schwann cells provide myelination to the cells outside the brain and spinal cord. So they do the same thing as the oligodendrocytes, but outside of the brain and spinal cord. And these glial cells are actually really, not only are they important for the brain's functioning, but they're also important when things go wrong. So you may have heard that, maybe there's someone you know where they get in a really bad accident and the brain swells. Well, why does the brain swell? Well, it's actually these glial cells. That's how they respond to injury. All glial cells, but especially the astrocytes, will swell after injury, which causes brain edema and causes that pressure that can cause injury. So it's not all good news. Also, these cells are more susceptible to tumors. So a lot of times brain tumors will actually be in the glial cells and not in neurons. So not all good news with these cells, but we have to have them. You wouldn't want to live life without them, but it's important to know both sides of them. They are susceptible to tumors, and they also are problematic in their response to injury. So here we see a couple oligodendrocytes, and again you see one covering multiple axons. And so here's the axon, and then here's what the myelin looks like, just covering all around the axon. And then this is a node of Ranvier. So this is the little part in between the two sheaths that's uncovered. So synapses, so those small separations between the axon and the dendrite, have three main components you need to know about. You have the presynaptic membrane that's up here, which is on the axon terminal. So right up here. You have postsynaptic membrane that's down here on the dendrite that's receiving the information. And then you have the synaptic cleft, which is the space in the middle that is empty, the gap. So we'll go back to that picture in a second, but a couple definitions yet. First, synaptic vesicles. These are small spheres in the presynaptic axon that contain neurotransmitters. So again, neurotransmitters are the chemicals that go across that synaptic cleft. So the vesicles are the little pods that contain these neurotransmitters and hold them until it's time for them to be dumped out into the synapse. So these dump out into the synapse, and this happens, as you see on the second point. The neurotransmitters are released in response to the electrical activity in the axon. So you have that signal come down the axon, which causes these chemical messengers to be released into the synapse, and then those connect with receptors on the postsynaptic membrane that are just specialized proteins that are designed to react to that neurotransmitter. So it connects, and it's much like a lock and a key. So the neurotransmitter is a key. It fits the lock, turns it, and activates that receptor, which then sends the signal down to the next neuron. So kind of going through the information here, you have both a real picture and an animation. So all this is called the presynaptic terminal. And again, presynaptic because it's before the synapse. Here you just have a mitochondrion, so the power plant. Here are those vesicles. So these are the little vesicles, the little pods that hold the neurotransmitter. Presynaptic membrane down here. And this is where the vesicles go down and dump out all the neurotransmitter that then goes into the synaptic cleft, which is this space in between. They connect to receptors that are on the postsynaptic membrane, and then the information is sent down the dendrite. Dendritic spines. These are studded areas on the dendrite that increase surface area. And what they basically do is they actually allow for more synaptic connections, more places where an axon can connect with that dendrite. And it actually is very important for neuroplasticity, as we'll talk about a lot more in coming lectures, because this is how they help axons find new dendrites and make new connections, which is important after a brain injury, or if you're training, trying to do something new, and the brain is changing itself with that. This is how they make those connections. The axon hillock, we'll look at this again later on. The axon hillock is a cone-shaped part at the bottom of the cell body that gives rise to the axon. So this is that part in the integration zone where all the information comes together. That happens in the axon hillock. An axon collateral is a branch of an axon that also ends in terminals and innervates other cells. So basically what will happen is a neuron will have just one axon, but they splinter into these little axon collaterals, and it enables the neuron to influence many different neurons. And lastly, axonal transport is the movement of materials within the axon. So what will happen is there are materials that need to be set up or down the axon, and we may need more neurotransmitters, so materials will get shifted up and down in order to make that happen. So axonal transport is just the movement of materials within an axon.